For this meeting, we are going to discuss the parts of the bacterial cell, and this is a topic that I am sure is just a review of your biology, your cell and molecular biology. So instead of looking into the bigger cell or the different types of plant cell or animal cell, we will just simply limit on what is expected of a bacterial cell. So in front of you is actually an illustration of a microorganism in which the different important parts are actually illustrated, and the microorganism is labeled. Um, of course, you have the different outer parts and going down to the deepest part of the cell. So in this structure, you have here the illustration in the microscope. Whereas in this structure, this is just a drawing or animated drawing of or illustration of a microbial cell. So you have here the parts, the capsule, the cytoplasm, the ribosome, the cell wall, the plasma membrane, the nuclear area, wherein you have the genetic property or the DNA, and then you have the plasmid. And for the outer part, you have also the capsule, the cell wall, followed by the plasma membrane, and then appendages like your fimbriae and the flagella. So we have to discuss first the different external part of the cell, or just simply the bacterial cell wall, which uh, is composed of the glycocalyx, the flagella, the axial filaments, and the fimbriae or even the pili. So based on the illustration, you can see here a cell in which you have here, it looks like a cell, but this is actually the flagella. So the flagella is shown in this manner, flagella for many flagellum. Flagellum is a singular one. So this is the structure of one flagellum. So this is actually anchored from the inner part of the cell, and then you have already here the main filament that make the composition of the flagella. So the flagella are found outside the cell wall. They are made up of flagellin, and they are actually attached to a protein hook towards the inner part of the cell, and they are anchored to the wall or membrane by the basal body, and they make their own rotation in order to aid movement of the cell. Um, we have here the bacterial glycocalyx. It is outside also the cell wall. They are usually slimy or even sticky. It is a capsule that is neatly organized that defines the shape or the movement of the microorganism. It has extracellular polysaccharide structure or chemical composition that allows the cell to be attached to the inner part or external part of the cell. So the capsule itself prevents phagocytosis by the presence of the glycocalyx. And then we have here an illustration of Streptococcus pneumonia capsule, as you can see here. So the different red shape is defined by the capsule of the Streptococcus pneumonia. And then you have here the slimy portion of the cell. So this is still the structure for flagella, as we have made mention. This is the basal body or the body of the microorganism itself. This is the structure that is made up of polymers that will provide us a hook and then you have here the flagella, structures of flagella. So this could propel in order to make movement of the microorganisms. However, there are different types of flagella. You have the monotrichous, which means it has only one flagella. We have also the amphitrichous. The amphitrichous has the flagella at its end of the microorganism. And then we have also Lopotrichous. It has several flagella at one side of the cell. And then for Peritrichus, the Peritrichus has flagella in all parts or in any parts of the cell. And then you have here an example of Monotrichous. How about this? You have the Amphitrichus. And then you have also here the Lopotrichous. And you have here the Peritrichus. So I hope you have remembered the microorganisms flagella. So the flagella moves in this manner: run, tumble, tumble, run, tumble, run, run. So in any way, it could start from run and then tumble, meaning to say, you just start to imagine the movement of an octopus. However, the octopus move against its filament. So this is actually running towards here. And then the flagella, if you notice, it spreads out. And then once it tumbles, it adheres to the surface of the organism or any other host. And if you notice, the flagella totally spread out. And that is the tumble. And then this will again close to its one another and then propel and then run and then tumble. So after the tumble, you have again the running. So the spreading and the closing and the propelling of the flagella makes the flagella move or makes the microorganism move from one place to another. So this is another structure showing to you the axial filament. So the inner part of the flagella could be shown with the presence of an axial filament. So this rotates. The rotation that happens inside propels the flagella itself. And then the axial filaments present in spiral cell. They are similar to the flagella, but they wrap around the cell. So example of this includes the Treponema Pallidum, the microorganism responsible for the disease or sexually transmitted disease, the syphilis. So this Treponema Pallidum are found in vaginal secretion. So they move with the moist. And then you have also another part of the cell or outer part of the cell, bacterial cell. You have the fimbriae or the pili. They are hair-like structure. The fimbriae allows attachment to teeth, stones in creek, plastic in catheters, hospital. So you could just simply try to imagine your own teeth could possibly have a microorganism that has this structure. And the fibrillar structure allows the microorganism to adhere to hard objects like our teeth, aided by the enamel of our teeth. Also likewise the stones in creek that makes it sometimes rough and slimy. and then also likewise in plastic catheter in hospital. And this could lead to the possible presence of infection if microorganisms are found in catheter. And then the pili are used to transfer DNA from one cell to another. How do they look like? The pili, the pimbre, and also the sex pili. They are actually very short appendages as compared to flagella. They are the it helps to adhere to the surface. That is one function, although they are very small. But their number is vast, so they enable to stick themselves to one place and also likewise move from one place to another. So the sex pili is actually an appendage of a bacterial cell that actually adheres from one cell to another, and that structure will form the bridge between two bacterial cells and enable the DNA to transfer from one cell to another, changing the character of the cell. So this is the one that I have just made mention. This is one cell. This is also another cell. You can see here the DNA structure. So you have here the 6 pili meeting the another cell. So this one will move through the 6 pili, the DNA, and will go here. So once the DNA is encoded in here, then it will form the trait that could be similar to this or form another set of uh, properties. So I repeat, the cell wall as part contains all those made mention, the pili, the pimbre, the flagella, and all other appendages. And these are actually anchored to what we call the cell wall. So the cell wall surrounds the plasma membrane, protecting the cell from osmotic pressure. And it has also the role of forming itself as a lattice uh, to protect other components of the cell. Another role of the cell wall is it prevents lysis, and they are made up of peptoglycans in the case of bacteria. So this is a typical chemical structure of a peptidoglycan. So from the word peptides, so they are made up of polymers of disaccharides. I think you can still remember your biochem. Disaccharides are actually carbohydrates, no? So they are made up of polymer of disaccharide and acetylglucosamine and N-acetylmuramic acid. So the NAG and the NAM made up the peptidoglycan, and they are linked together by, of course, the polypeptide. So the violet here, the violet ball, represents the NAG or the N-acetylglucosamine, and then the green one are the NAM or the acetylmuramic acid, N-acetylmuramic acid. And then the red one that link together the violet, the NAG and the NAG and the NAM is a red structure, and that is what you call the peptide brins. So they are linked together by a polypeptide. So you have here the polypeptide, the peptidoglycan. So this is just a illustration how the NAG and the NAM works with the peptide bond. So these are the NAG and the NAM, and of course you have here the peptide bonds, no? And these peptide bonds will link together with the structure of the tetrapeptide side chain. So, ito yung green, ito yung peptide glucose bridge. Yan. And then this cylindrical shape here refers to your NAG and the NAM. So what binds them together or links them together is the presence of these tetrapeptide side chains and intertwined with the peptide cross bridge. And then this could be illustrated in a gram-positive microorganism. Uh, the rough portion there is your peptidoglycan. However, this could be illustrated in this manner. So these are your peptides, and they are made up of the cell membrane. So this cell membrane here protects the peptidoglycans, also illustrated here. Okay, this is an illustration of a gram-positive SRUs. So the peptidoglycan is inside each individual spherical structure that you could see. And of course, yung shape is defined by the cell wall. Okay. And then you have the gram-negative cell. They are thin, poorly organized peptidoglycan. That is why the outer membrane, lipoprotein, lipopolysaccharide, phospholipid, or LPS, uh, protects against phagocytosis. Uh, gram-negative cells are usually the properties of gram-negative cells are usually illustrated by the penicillin. So this is your gram-negative. So last time the peptidoglycan, the violet one, is not covered by two outer membrane but only one membrane. That is for the gram-positive. But for the gram-negative, they are both covered in the same area. So this is your gram-positive. This is your gram-negative. If you consider this portion, it is covered by two yellow layers. Whereas in this case, it has only one yellow layer. So it is hard to color this one. This one is easier to color. So this is the other illustration. So eto parang cake lang na may icing sa ibabaw. But with the case of gram-negative, parang sandwich na nakatago yung mga numb and the numb. This is the gram-negative cell wall. Ito yung sinasabi ko na naka-sandwich lang yung numb and the nug. Siyempre, ito yung mga polypeptide bonds covering or providing a sandwich to sandwich the numb and the nug. Okay, this is an example of a gram-negative microorganism, the Escherichia coli. Once stained with gram stains. We call it gram stains. The color is red. Pero yung gram positive, the color is violet. So the gram positive is violet because it absorbs the dye or the stain that is used in defining its structure. Whereas for the case of the gram negative, uh, red lang siya, no? So this is the structure of a gram-negative outer membrane. It is made up of lipopolysaccharides, lipoprotein, and phospholipids. It forms the periplasm between the outer membrane and the plasma membrane, the one that I was telling you as a sandwich. Then protection for phagocytes, complement, and antibiotics. So because it is highly dense. It prevents some phagocytes and antibiotics. And then the example of this is yung character trait of Escherichia coli. The lipid A is an endotoxin to gram-negative outer membrane of gram-negative, forms a form of protein formed channels through the membrane. Okay, here is an illustration to distinguish between a gram-positive cell and a gram-negative cell. So, the peptidoglycan of gram-positive is thick, whereas the peptidoglycan for gram-negative is thin. Easy to memorize. Yung negative is thin. Yung positive, mas makapal, syempre, kesa yung negative. And then, the gram-negative has teichoic acid. Gram-negative, negative din siya sa teichoic acid. And then, it contains mycolic acid. But in the case of gram-negative, it has an outer membrane. Okay. And then in terms of cell wall, um, microorganisms contains mycoplasma, or there are some microorganisms like mycoplasma and they lack cell wall. And then for the case of archaea, they have a wall that is lesser in shape or in size. There are some microorganisms that contains sterol in the plasma membrane, whereas there are some microorganisms that only contain the pseudomorane, or the cell will do not have the NAM and the delta amino acids. Okay, let's assume the grades peptidoglycan versus penicillin and other antibiotics affects the synthesis of cell wall. And then this could be illustrated here. You have here the plasma membrane in cell. So the plasma membrane could be destroyed by the presence of other noxious substances like penicillin or related antibiotics. So this is again the chemical structure of your plasma membrane. You have the phospholipid bilayer, the phospholipid bilayer, and then you have the peripheral protein. The peripheral protein are the blue ones scattered around. And then you have also the phospholipids that rotates and move laterally. So, eto yung mga phospholipids.